Okay, guys, I am going to apologize profusely for having to present this lecture via video, but yeah, the entire house has been wiped out with pink eye again for the second time this month. So I'm keeping my distance. I'm going to stay away from you guys. I do not want to spread this. This is awful. But the show must go on as we are getting closer to the end of the semester. And I think we have had so much sickness in this house because we have moved back from a pretty isolated part of California to a major metropolitan area. And I think we are now revamping our immune systems to try to deal with all the bugs and the illnesses that have been circulating in such a condensed area. So I hope this is just reacclimation to civilization, and I hope we are done with this until next year. Anyways, I just kind of want to refresh your memory on where we left off, and we're going to continue this lecture and finish it, and then I'm going to start a second one, as I probably will not be in on Friday as well because I'm still contagious. So we have been talking about the possibility of muscle releasing a growth factor during contraction, and that growth factor is going to have a pretty profound impact on the microvascular system. And when I say microvascular system, I'm of course referring to the capillaries and the small arterioles that are feeding into the capillaries. So again, this is another example of local control, but this is local control that's going to have pretty favorable adaptations that are coming from skeletal muscle contraction. So we kind of left off on this slide, and I was showing you that when a muscle contracts, it can release this growth factor. So that would therefore make skeletal muscle an endocrine organ, right? So it can secrete messages. And it's going to secrete this growth factor. This growth factor is therefore going to bind to its receptor. On this picture here, we have VEGFR2 receptor, which means vascular endothelial cell growth factor receptor 2. It's just so much easier to say VEGFR2. And then once this ligand or this growth factor binds to its receptor, we are going to have one of the first initiatives to cause favorable adaptations to this vascular system here. But again, as we mentioned before, there are several other factors that are inducing this. One is the constant release of eNOS, right? Or release of nitric oxide as a result of eNOS activation. So eNOS, in combination with increases in shear stress, in combination with stretching of the endothelial cells, right? Because there's a mechanical and a chemical stimulus that will cause these adaptations to occur. All of these factors will induce changes to the vascular system that is localized by the skeletal muscle. And of course, since we're talking about muscle in this class, we're talking about muscle mediation and how muscle can initiate that signal. We did go over the cell signaling process. Let me do it one more time for you, just in case the animations threw you off a little bit. If we were to have VEGF circulating, and in class we asked, well, where does VEGF come from? Well, it comes from skeletal muscle, but there's also other cells that release VEGF. Endothelial cells, which we're talking about right now, that line the inside of the vascular system. So let me go back here. We know that within this vascular system here, we have endothelial cells that are complete. Here, let me see if I can add them just to really knock this thing out of the park. Let me see if there are any. I do not see any shapes, but I'll just draw some dots, right? So we know inside here we have endothelial cells. There's 1, there's 2, there's 3, right? We have them on the bottom as well. 1, 2, 3. We'll put some nuclei there, right? So dot, 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 and this occurs all throughout the capillaries, right? And these endothelial cells, they are sensitive to both gas, right? They're sensitive to nitric oxide, right? We guys drew that for me on the last exam, where when nitric oxide is released and created in the endothelial cell, that will travel down into the smooth muscles, which are right about there, and that will cause relaxation of those smooth muscles, right? But they're also sensitive to chemical signals because, of course, let me zoom in here. There are receptors that are, look at my little Mickey Mouse hand, right? There are receptors that are on each one of these endothelial cells, and those receptors, of course, can allow VEGF to bind to them, right? So I'll just draw a receptor here, right? So endothelial cells can receive the message from the skeletal muscle and they can initiate a response. But again, this is a combined effort, right? It's mechanical stretch to the endothelial cells. Where's that mechanical stretch coming from? Well, it's coming from shear stress, right? We have this increased pressure that is coming through the microvascular system. We have nitric oxide being released, and we have this chemical messenger. So when we come back here, oops, sorry about that. This membrane right here, this membrane is an endothelial cell. So I'm just going to put EC, right? And then on the outside of the endothelial cell, we have blood, shear stress, right? Shear stress, we have red blood cells, right? Red blood cells. And then we have VEGF, which is a chemical messenger that can bind to these receptors and initiate a response in those endothelial cells. So once we have the secretion of these chemical promoters, they are going to bind to their receptors. And as we mentioned earlier on in our presentation, at first these receptors exist in a monomer form. So VEGF has to bind to the monomers. And then once these two bind, we are going to have dimerization of this receptor, and that activates the receptor. And once it's active, we have phosphates that are essentially going to bind to the tyrosine. I'll just put TRY. That looks awful. But the tyrosine residues, and tyrosine, of course, is an amino acid, right? So once this receptor becomes active, it is going to recruit a protein. This protein has the name of PLC gamma. PLC gamma is going to take one of those phosphates and it's going to deliver it to this membrane-bound protein called PIP2. And usually membrane-bound proteins are bound via another protein which stabilizes it in the membrane. And it's going to essentially hand off a phosphate. Now, once PIP2 becomes active, because it's phosphorylated, it is going to hand off a phosphate to DAG, which is diacylglycerol, which is just a type of fatty acid. And of course, this protein is also anchored into the membrane, right? So that phosphate gets handed off, and then this will recruit another protein, which is IP3, and IP3 is going to translocate here to the endoplasmic reticulum. It's not a sarcoplasmic reticulum, it's endo. I'm just going to put ER, endoplasmic reticulum. And once it binds to the endoplasmic reticulum, we're going to have a release of calcium, right? Calcium is then going to bind to calmodulin, right? This is a calcium calmodulin complex. So a CCC, right? Calcium calmodulin complex. And this complex is going to do something very unique to this protein here, which is NFAT. Now, this protein is inactive when a phosphate is on it, right? So some proteins operate that way, right? When a dimerized receptor becomes active, it gets phosphates. But this protein is unique. The NFAT protein has to be dephosphorylated to become active. So that phosphate has to be removed. And then this is active, and this is a transcription factor. So this is going to translocate to the nuclei, right? So if I have the nucleus here, it's going to translocate. It's going to bind to DNA. So calmodulin complex removes that phosphate. NFAT becomes active, right? So we'll put an exclamation point. It's turned on. Once it's turned on, it will translocate and bind to DNA. And here it will start to transcribe and translate for proteins. So this is how a protein is put into the cytosol of the cell. Now, in this particular case, this is going to transcribe for COX-2 mediated proteins, which the one that we're most interested in here is the prostacyclin. And we know this is a vasodilator, right? So VEGF signaling can induce vasodilation, which is really important for adaptations to the endothelial cells, right? We need dilation, we need migration, and we need proliferation, okay? So DMP, dilation, migration, proliferation. And if we go back to a slide I showed you earlier, let's just go back here. This portion here is a normal endothelial cell layout, right? But when we get VEGF signaling, right? So we get VEGF signaling and we get prostacyclin. I'm just going to put PRC, right? And we get mechanical stress. Well, that causes changes in that endothelial cell complex, right? We no longer get this nice, clean, organized endothelial cell complex because they start to migrate and they start to proliferate. And again, when I told you guys about migration, I told you guys to think about the geese that fly in a V, right? Well, those geese as a unit, they're moving, right? And for the sake of migration patterns, they're moving south where it's warmer. These endothelial cells, they will start to migrate and they open this lumen wider, right? They will also start to proliferate, and that means they will start to essentially take one cell and turn into two, right? So one cell, the cells will divide, right? And that's proliferation. One cell will turn into two, and that's how we get this kind of vascular change, right? So VEGF is capable of doing that. Well, let's go back to this. VEGF, mechanical stretching, nitric oxide, and changes in shear stress all kind of contribute to those changes. Okay, so now the second pathway that we talked about in class that I'm just going to kind of revisit here, this is one that is focusing on migration and proliferation. So again, migration is going to be cell movement and proliferation is going to be cell division, right? Both which are really important. And in a moment, I'm going to show you a video that will show you proliferation and migration that is induced by VEGF. So let's just kind of go through this one again really quickly. Now, this is the same initiation. So we have the endothelial cell right here, right? This is the membrane of the endothelial cell, and then floating here in the circulation, we have red blood cells, right? Here's a red blood cell, here's a red blood cell, and we have these growth factors circulating in here. Sorry about that. We know that VEGF, that it was secreted by the muscle, is going to bind to the monomer receptors. The monomers are going to create a dimer. They are going to autophosphorylate on those tyrosine receptors. And again, tyrosine is just an amino acid. It is going to recruit the PLC gamma protein, which is then going to interact with PIP2, delivering a phosphate, and PIP2 is going to give that phosphate to DAG2. Again, these are anchored into the membrane, so these don't move. Now, DAG is going to recruit some different intermediates than we saw with the prostacyclin or vasodilator pathway that was on the previous slide. So this one is going to recruit a series of enzymes. This is called PKC, protein kinase C. Protein kinase C is going to recruit RAF1. RAF1 is going to recruit something called MEK, M-E-K. And then MEK is going to recruit something called ERK. Now, importantly, this exact same signaling pathway using these mediators, which is also called the MAP kinase pathway. These are the proteins that are upregulated and hijacked when cancer is present. Why are they upregulated? Because the cancer cells want to induce angiogenesis and arteriogenesis as quickly as they can. One of the reasons being is because they'll use those vascular tubes to metastasize and spread. The other reason is they want to increase the delivery of nutrients as quickly as possible because cancer cells rely primarily on anaerobic metabolism. They don't use oxygen. They don't use a lot of fatty acids. So they're going to have more glucose delivered as quickly as it can be through angiogenesis. So interestingly, exercise increases this same pathway, right? Now, ERK is going to translocate to the nucleus of the cell, right? So here's the nucleus, and inside it's the DNA. And once ERK1/2 translocates, this cell will listen to VEGF's instructions, right? VEGF has the instructions. These mediators read the instructions and translated the instructions. And then ultimately, ERK tells the DNA, you need to proliferate and you need to migrate. So what happens is we have one cell turns into two cells, right? So there's the proliferation, and we get some movement of the cells as well. So those are the two pathways. One would be dilation. The second one, which is the one we're talking about here. So here, this first one is dilation, and this is mediated through prostacyclins and calcium. And then the other two are migration and proliferation. So again, we're talking about dilation, migration, proliferation. And just so you can see it, DMP. That is how we create new vessels. Dilation, migration, proliferation. So now here, we're just going to kind of review that arteriogenesis is this widening of the lumen based upon primarily, and it's not 100%, but these mechanical stressors, right? Muscle contraction, releasing VEGF, changes in cardiac output, right? Changes in Q, increases in shear stress. This would also be increases in sympathetic nervous drive or sympathetic nervous system drive, right? Because we're going to have more dilation of the arterioles, right? Increasing shear stress. And also changes in O2, in this case, low O2, right? Because as we're exercising, there's always kind of an oxygen deficit, which is what leads to ischemia. And this is going to lead to widening of that lumen, right? More mechanical than anything. And if you want another picture that just kind of does a better job of it, this is before exercise. And this is when we've been exercising for a long period of time. The lumen of the vessel, so if I draw the vessel like this, right? The lumen of the vessel becomes wider as a result of that change in mechanical stress, which is being delivered more frequently as a result of exercise. Now, when we talk about angiogenesis, we're talking about more of a chemical mediation. Now, that's not to say contraction, heart rate, shear stress, and oxygen doesn't play a role. They do. They all play a role, right? When you have more regulators involved in growth, it makes it harder for diseases to figure out the synergy between all of these things, right? It makes it harder for diseases such as cancer to hijack certain systems because there's multiple regulators. And what we see with VEGF is we don't see a widening of the lumen, right? We don't necessarily see this occurring, but what we see is a new vessel being created. And that new vessel starts from generally the arterial, right? We have the arterial, it could be an aorta, it could be a arterial vessel that is leading to skeletal muscle. We begin to get this sprouting, which you can see here. And then that sprouting turns into branching. And then that branching increases capillary density. And now we have many favorable adaptations because of this, and we will talk about this momentarily. So this is something that we refer to as vascular remodeling. And vascular remodeling is a combination of both arteriogenesis and angiogenesis, right? And this one seems to be more driven by chemical messengers, where this one is more driven by changes in shear stress and mechanical stretch or mechanical stress. Okay, so this is something you've already seen. I'm just kind of beating a dead horse. I'm telling you what oxygen does to kind of create these changes. And, you know, we talked about oxygen demand, right? We said that we kind of limit our oxygen intake with exercise. So there's an increased demand for oxygen. Shear stress. There's another growth factor called hypoxia-inducible factor. And this is basically the cousin of VEGF. So when there's low levels of oxygen, as we talked about here, right, we have ischemia or low levels of oxygen, that will upregulate another growth factor called HIF or hypoxia-inducible factor, which does the same thing as the vascular endothelial cell growth factor, right? So those two are cousins. And then there's another cousin that we're going to talk about momentarily on the next slide that does a very similar thing. So there's three kind of growth factor cousins that will all induce vascular remodeling. Now, the one that I have here that's in blue, this is really important because this is inflammation. Inflammation is going to play a very important role in vascular remodeling, and inflammation is going to recruit something called MMP. And if you haven't heard of MMP, MMP is a very specific type of protease, and I want you to think of an MMP as a pair of scissors, or think of MMP as Pac-Man, right? Because I can't draw scissors using my here. He's a happy Pac-Man. He's smiling, and let's say this Pac-Man has a hat, just because I feel like him wearing a hat. There you go. MMPs are going to be very important in assisting with vascular remodeling, and I'm going to draw you a picture of that momentarily, okay? So I'm just beating a dead horse here, and now I'm telling you that inflammation and the recruitment of these proteases are also going to play a role in vascular remodeling. So on this slide here, I'm just kind of putting it all together. I'm saying, hey, these are pretty important inducible factors. And if you don't remember what they are, I put a definition there. So we have shear stress. We have this mechanical stretch, right? I told you endothelial cells are sensitive to stretch. And I also had showed you a picture right here showing you that endothelial cells are sensitive to stretch, right? Which is why changes in shear stress are so important. Let me get back to this slide. So we talked about eNOS and nitric oxide. We talked about VEGF. This is PGIF, which is placental growth factor. And this increases vessel enlargement as well. So we have VEGF. We have hypoxia-inducing factor, right? We had that on the last slide. And then we have this PGIF. And the only reason I brought up PGIF is because if you look here, PGIF is a growth factor that can use VEGF receptors, right? So there's a couple of these growth factors circulating. And then lastly, this is MMPs. And if you don't know what an MMP is, it's a membrane, right? Here's the first M, metalloproteinase. Okay, MMP. And this is instrumental in remodeling the extracellular matrix. And I will tell you why we need that to remodel capillaries. And this needs inflammation. So MMP and inflammation, they're kind of working side by side to get the job done. All right, so I want to show you a video of this very same mechanism occurring that was captured using microscopy. They had to magnify this several hundred thousands of times so that you can see it. But here we have a vessel. This vessel, I believe, is an arterial. And we are going to start seeing some sprouting from the arterial as a result of VEGF signaling. So you can start to see some sprouting here already. You could start to see some sprouting here. But I want you to take a look at this and just kind of watch how this... let me turn off the volume. How this works. So here we go. Here you can see the sprouting, and then you can start to see the branching. You can start to see it all across the vessel here. You can see some of this globulin proteins down there, which are MMPs. You'll probably see another MMP up here as well. So watch. It's going to be right there. So there's an MMP. So an MMP is responsible for essentially cleaning up the extracellular matrix. Okay. Now what I want to show you here is VEGF-induced migration and proliferation. So here we have an endothelial cell in culture. Here we have an endothelial cell in cell culture. They basically put a bunch of VEGF in the medium that the cell culture is in, and watch how they will start to move and proliferate and divide as a result of VEGF being in this medium. So let's take a look at this. And you will see that these will come together, these cells, they'll create a lumen, so new blood can be delivered through that lumen. So look at, you can start to see a new vascular system being created here, and here's the lumen where blood can travel through. And that was from 2 endothelial cells that were fed VEGF. Now this is just a fluorescent histochemistry stain that just is kind of showing you where one cell was and where the other cell was and how they kind of came about. And you can see that a lumen has been created here. And that is essentially how these cells are responding to those growth factors. Okay, so now we're getting to the specifics of capillary or vascular remodeling. And there's 2 types of remodeling that we're going to talk about. And I have a picture here that was in your paper that you read on how vessels grow. And we kind of talked about sprouting, and we talked about hollowing, right? So when I talk about hollowing, we're talking about the angiogenesis or the arterial genesis, right? We see that we have this vessel and it becomes more hollow, right? So we start to lose a little bit of the basement membrane and the material that is surrounding where the endothelial cells are, right? So that's what we're going to refer to this as, both sprouting and hollowing. Now, the first one that we're going to talk about is this process called intussusception. And I put the enunciation here because it's really difficult to say. So what happens when we have this mechanism occur, basically we get one vessel that will basically divide into a second vessel, and they call this invagination, right? So you can see that VEGF is capable of taking a existing vessel and basically through signaling, through migration, and through proliferation, we will start to assemble a new divider here, right? So one vessel will turn into 2 vessels, right? They both share the same initiating vessel, which is here, right? But the VEGF signaling and the proliferation and the division of the cells and the migration of the cells have caused a reordering of those endothelial cells to create 2 independent vessels. And again, this is called intussusception. Now, this particular type of vascular remodeling, here's all the stimuli, seems to respond most favorably to stretch of the cells. So mechanical signaling, right? Increases in cardiac output, increases in shear stress, and low levels of oxygen. So they believe that this type of remodeling is more favored. It favors more of these mechanical stimuli. Okay, which is not to say that VEGF doesn't play a role, but they believe that the increase in surface area that comes from this type of remodeling it's more sensitive to mechanical stimuli. Okay, now when we get on the other side of things, I showed you this picture before. Another thing that intussusception does is it increases hollowing. Right? Let me change the pen color so I can kind of. Where is it? Oh, I don't know where it's at. We're just going to keep. So it also creates new vessels going side by side. So if we have two arterioles that are running parallel to one another, this type of change will increase the lumen size, as you can see here, right? We have a wider hole, and it also will cause a little bit of branching where we'll have endothelial cells and lumens being created. Think of that video I just showed you, which will basically allow blood to be delivered to parallel vessels or parallel arterioles, right? So this is how mechanical changes and mechanical stress impact larger vessels, right? Now the other one, which is sprouting, this one is primarily sensitive to VEGF and chemicals. So here we have a capillary. You can begin to see some sprouting here, right? Gets a little larger, right? And then we have a whole new tube that is created. And this one is primarily driven by a chemical signal. And then what we can see here is before VEGF and exercise and increases in mechanical stimuli. And then here we see after. And you can see that the capillary density on this side, right? So these are all skeletal muscle cells, right? And then you can see interstitial space here in the white, right? That is where VEGF is going to accumulate in contracting muscles. It's going to accumulate in the interstitium, right? But you can see that we have way more capillary density here surrounding each one of the fibers. So, what is that going to do? It's going to improve oxygen delivery, better nutrient supply, most importantly, waste removal, thermoregulation. Well, what happens when we have more capillaries? Well, we have greater release of heat. So heat is not accumulated in the muscle because we know that heat can change things in the muscle quite rapidly and decrease performance. And also we will have enhanced endothelial cell function with more capillaries. And then the last thing I want to talk about, and I will draw a picture on this momentarily, is we are going to talk about this extracellular matrix degradation, right? And the inflammatory process and the response of MMPs. So I'm going to start by kind of showing you the inside of the lumen, right? So here is the vessel, right? If we're talking about capillaries, we know that endothelial cells line the vessels. You guys have drawn that for me. We know that we have smooth muscle cells here, right, which receive the nitric oxide. And then we have some other kind of fibrous tissues here. Well, if we look at how endothelial cells are, how they're connected to these other tissues, they're connected and they're anchored. So here that endothelial cell is there, it's anchored. Here's an endothelial cell here, it's anchored. Here's an endothelial cell here, it's anchored. Here's an endothelial cell here. It's anchored in this tissue right here. Well, they're also anchored side by side. So here, here, and here. So these anchors are called adhesion molecules. Okay, so if I drew an endothelial cell like this, right? Here's one endothelial cell. And then here's a second endothelial cell. There's a little bit of space between them. Well, also, if we look at this tunica tissue here, right, we'll just say that that's right here. These cells have to be anchored to that tunica as well. So we're going to have these anchors here, anchor here, right? Here is the nuclei of the endothelial cell. Here is the nuclei as well, right? Here's the receptor for VEGF. Here's the receptor for VEGF, right? Everything's here. Well, there's also adhesion molecules that go this way, and they anchor the endothelial cells to each other. Okay, so let me just kind of color that in a little bit. Just say that these adhesion molecules are superglue, or they are anchors that keep the endothelial cells bound to the tunica. and also bound to each other. Why do they have to be bound? Well, because these things are they're directly interacting with shear stress, right? So if blood is coming through and pressing on these things, well, what's keeping them from just being pushed away and flushed away? Well, it's because they're anchored. Now, when I told you earlier that these MMPs are like scissors or Pac-Man, well, when we have cells being told to migrate, how do they migrate when they're held down by these anchors? Well, that's where MMPs will come in and be in like scissors. This is one side of the scissor, right? Here's a second side of the scissor. Thank God I didn't go to school to be an artist, right? Here's scissors, handles, right? Well, the MMPs are going to chop these adhesion molecules up so that endothelial cells can migrate and move, just like you saw in that video. Without MMPs, membrane metalloproteinases, these cells can't proliferate and they can't migrate. So this is why these adhesion molecules have to be cleaved so that the endothelial cells can move. Okay, that's all I have for the lecture part. I'm going to come back and draw in a moment, and then this one will be in the books. Okay, so let's go over a little bit of the image that I had on the board for you in class, and let's kind of talk about this. So I'm going to be zooming in and out, and we're going to kind of talk about everything that we have seen so far. And let's start here with the arterioles, which we also call resistant vessels. And they're resistant vessels because, of course, they are, let me get this thing working here, they are receiving signals from the sympathetic nervous system to essentially, let's do blue, to essentially contract, right? So we are getting... here's the sympathetic nervous system, and I have it right here for you, right? And they are releasing catecholamines, and those catecholamines are binding to receptors, and they're essentially telling these are arterioles or resistant vessels to constrict, right? And we know that's the sympathetic nervous system's job is to constrict, and that is going to increase pressure and increase shear stress. Well, that's a positive thing, and we know that now because we know that endothelial cells, as this shear stress gets more intense, endothelial cells are going to respond to that, right? Now, beneath that, we have skeletal muscle, and we know that skeletal muscle contracting is going to release VEGF, as you can see here, and that VEGF is going to be in the interstitium, which is the interstitial space between our microfibrils, right? So if we have these skeletal muscle cells, the myofibrils, sorry, we have them like this, right? And the myofibrils are stored like that. Well, all of this space, let's just do blue, all of this space here, that is the interstitium. Clean this up, right? And all of this space here between the fibrils are going to be where the VEGF is going to be collected, right? And we know that VEGF, since it's coming from contracting skeletal muscle, there's going to be a concentration gradient where it's going to move from high, right? Obviously, we're going to have a lot of, let me change that here. We're going to have a lot of VEGF in the interstitium, right? And that is going to be able to interact with endothelial cells. So let's go over this. So inside of the circulation, let me go back to red. We have red blood cells, right? And we also have, I'm just going to make VEGF being as these circles here, right? We have these growth factors, right? We also have the, what was the other one that I had mentioned to you guys? Do you remember the cousins that I was telling you about, right? We have, let's say PG, right? That was, I'm sorry, the PIGF, right? The placenta growth factor. And then we also have, let me get another one here. We also have, what was the other cousin that I had mentioned? It started with an H. Do you guys remember that? HIF, hypoxia-induced factor, right? So we'll have that kind of circulating in here as well. And all I'm just trying to show you is that we have these growth factors that are responding to exercise. They're responding to all the stimuli that I was talking about, and they're being secreted, right? Now, once we, let me change back to black. Once we leave the arterioles, right, and we start to get into side of the capillaries, which I've labeled for you here, right? Obviously, this would be the oxidative side, right? And this would be the deoxygenated side, right? So we have oxygenated blood here, and then deoxygenated blood here. All of it's dropping off oxygen. I did this particular path to kind of zoom in, right? And I said here that this is a zoomed in capillary branch. And what you see here is endothelial cells, right? Let me make this a little thinner. Okay, so we have endothelial cell, right? We have another one here, we have another one here. And then we have these endothelial cells here that have receptors on them. So a couple of things are going to occur. When we have VEGF, so let me go back to orange. When we have VEGF bind to their ligands, okay, you see that? These are all VEGFs. We're going to have a couple of things happen, right? And as the video showed you, one of the things that's going to happen is sprouting, okay? So how does sprouting happen? Well, it could happen a couple of different ways. We can have this particular cell start to undergo proliferation, right? And what happens then is when that cell divides, we will get a new cell here. And then if VEGF continues to signal to these cells, we'll get more endothelial cells. You see that? And then we'll get more endothelial cells. And then we'll get more endothelial cells. Now, this is an example of sprouting. These are all new cells that will eventually turn into branching, right? So the cells are being told to proliferate, divide, divide, divide. And then what happens is we have an entirely new vessel that is being created within an existing vessel, right? And what happens then is as these branch out more, they will, just like the video I showed you, they will create a lumen. And you see how these become hollowed out now? Let me make that a little smaller because I'm erasing everything I just did to, right? So look at this. They start to become hollowed out, right? And now, back to this just to clean it up. Now we have a new vessel. So that is one way VEGF will signal to proliferate, right? Now I'm going to zoom out a little bit and we're going to go down to these cells over here and I'm going to, let me draw one more. That's not what I wanted. Let me draw one more cell here, right? Okay, and I'm going to number these. This is going to be 1, 2, 3, and 4. You guys see that? Okay, so another way VEGF can cause angiogenesis, right? Here's our VEGF molecules, or our VEGF growth factors. They are going to bind to those 4 cells. Now, this will be a signal for migration, right? Not proliferation. We're not dividing. We're going to have these cells move. So watch what happens when these cells migrate. Well, we're going to get a very similar thing. We're going to have the cells begin to move, right? They're migrating. Think of the geese, right? What do the geese do? They begin to migrate. And here's what happens. So this one was number 1. So now number 1 migrated here. This one was number 2. Number 2 migrated here. This one was number 3. Number 3 migrated here. This one was number 4, and number 4 migrated here. So they all moved because of the VEGF signal, okay? Now, one of the things that begins to happen is, let's create a lumen, right? Because the video showed us that once these things move, they generate a lumen. So we now have a lumen, right? A hollowing being created. Okay, let me erase number 2 here. We know these cells migrated, and now they're hollow inside. Well, if VEGF keeps signaling, right, because these are endothelial cells, which means they have receptors, right? So they all have VEGF receptors, right? If VEGF continues to signal, well, we're going to have more proliferation, and then we're going to have more migration, right? So what begins to happen here, let me just clean this up a little bit so that we can continue drawing here. What begins to happen now is the signal for migration and proliferation come together. So we have proliferation, we might have migration, and look at what happens. These 2 different sprouts start to make branches, and now they are another capillary network that has lumens capable of delivering blood through. And that is how that works, okay? So VEGF is secreted from the muscle. VEGF travels through the capillaries. VEGF will signal the endothelial cells, right? The endothelial cells that have these receptors, and it will cause proliferation and migration of existing cells. And now blood is capable of traveling. Let's change the color to red, right? Red blood cells are now capable of traveling through these new vessels and drop off oxygen, take up CO2, right? We can have more, I'll make it green, we can have more glucose delivered to the contracting muscles, right? And that's how that all works. And as we saw, let me clean that up. As we saw, other things that are occurring as well, if we move over here, right? If we move over here to these parts of the capillaries, we can also have the creation of new vessels from existing vessels, right? So we talked about this word, right? We talked about intussusception, and basically what is going to happen there is we are going to have division, right? Division of an existing vessel, which will ultimately lead to 2 different vessels, right? So we will now have blood being delivered that way, blood being delivered that way. These are 2 different vessels that now have this kind of barrier between them, and this barrier has come about because of endothelial cells that are migrating and moving, and eventually we'll get I'm just trying to do all this multitask. We'll get a vessel going this way and a vessel going this way, and they will have their own blood delivery capacity, right? So that will eventually look like this. And I can get rid of this and get rid of this. and then get rid of this. Right? And then what we saw there as well is that we can also get some networking between, which would look like this. Right? So that is an overview of how all of this kind of functions together. Okay? I really hope that helped you kind of visualize and see, especially this part here, because when we get into the cellular stuff, it's so neat. I really hope that helps you kind of see how this is occurring. Now, the other thing I'm going to show you is how MMPs play a role in this.